Welcome to my online talk at this year's Nextflow Summit. I'm really happy to be here and present the work that I've been doing within the Germany-wide project PM4Onco and NF-Core. And just as a heads up, with this talk, I want to motivate others to think about ways to integrate contributions to open source community projects such as NF-Core into their own research work, no matter how big the project might be. I'm Famke. I'm a PhD student at the University Clinic in Tübingen. I work both at the Translational Bioinformatics Group and the Quantitative Biology Center. So a quick agenda before we get started. I will introduce you to the PM4Onco project first, and then I'm going to tell you a bit about how we looked for needs and requirements within this bigger scope project. I'm going to introduce you to our concept of a reference pipeline, and then also how we got involved with the NF-Core community. And then in the end, I'm going to show you a few things that we recently added to a very popular pipeline, and so what we accomplished in that sense. The PM4Onco project, which is short for Personalized Medicine for Oncology, is one of the medical informatics projects which reside within a Germany-wide project that aim to connect efforts made in the last few years within the medical informatics initiative and then bring these into clinical practice. And in our case, this is with the focus on cancer patients, but there's also similar projects with other diseases in focus. And you can see here on the slide a really nice overview how the project is structured, right? So we have 6 different work packages that are working on different aspects that come into play when you think about personalized medicine for oncology. So we have one work package working with cancer registries. We have a work package that is defining a core data set to be used for data exchange. There are people working on patient-reported outcomes, visual analysis, and advanced analytics. And then there's also my work package 4, reproducible bioinformatics, which you can see it's mine because it has this red box around it. And all of these 6 work packages are in close contact with the cancer registries and the data integration centers at the clinics. The data integration centers are centers to collect and anonymously store data produced within the clinics. And we also have these molecular tumor boards, which have been established in the last few years, where a combination of different experts, so clinicians, bioinformaticians, and so on, come together and discuss different treatment plans for individual cancer patients. So these are our interactions. And then we have a work package 7 and 8 who are doing the rollout and the coordination of the whole project. So you can see this is really one building block of a bigger project, really big research project, and also application project within Germany. And each of these work packages has their own set of goals who are being defined. And I'm just going to show you one of those goals, which is related to the work that I'm presenting here. So that's why I'm showing it. So one of the goals of work package 4 is to define a reference bioinformatics pipeline. And I put into brackets here for variant calling. So just a quick reminder or information, variant calling is the process where you find different regions or even only bases within the genome that differ from a reference. And in our case, we're looking for these kinds of differences within tumor samples. So part of these goals are the establishment of metrics for comparing such pipelines, conducting benchmarks to compare them, tool development, so tools that can be integrated in pipelines like this, and also statistical evaluation in the end, and workflows that go towards therapy recommendations, so towards the molecular tumor board. Within this big project, there's a lot of different hospitals who are part of the big project. These are scattered across Germany, as in probably most countries. And we know that most of them use any type of bioinformatic analysis to do the care, right? So some of them might use commercially available tools such as Illumina Dragon or Kiagen for the variant calling. Some of them might use an in-house pipeline, and some of them might use open source available pipelines. And what we aim for is to have all patients receive the same sort of quality of care in all treatments. And thus we have to agree on certain best practices for the data analysis, from which one step is the variant calling. And that's why we want to establish this reference pipeline. And in order to figure out what are the needs, what do people want in this sort of pipeline, we did a survey where we had a big table in the end, and we also started working with the benchmarking platform MC Bench, which was previously only able to show you how well your variant calling pipeline performs in regard to germline variant calling. And we, within this project, we expanded this to also be able to evaluate somatic variant calls. And just I want to show you quickly our benchmarking study. This is a work in progress where I'm also working on a manuscript, so keep your eyes open if you're interested in something like that. But we gave 2 different data sets, which are FastView files, basically, to the different university clinics, let them run their own pipelines on these files, upload the results, so the BCF files, to a OSF project. And then we ran the benchmarking workflow on these results. And now we're still analyzing the tables full of false positives and false negatives to understand what type of tools work best in which settings. But going towards the reference pipeline, this is not only something where we care about which tool performs the best, but we have also other requirements. For example, we want this reference pipeline to be portable and reusable, because we want to roll it out, right? So we want to allow people to compare their own results to the results of this reference pipeline, but we also want people to be able to just use it if they don't have the time to develop something themselves. And this is nicely solved by Nextflow. I don't think I need to explain benefits of a workflow manager here, but not only do we want this pipeline to be portable, reusable, and working on different platforms, we also need it to be extendable, have version control, and long-term availability. Because as you know, PhD projects are notorious to die if they're not part of a bigger scope project. That's why we are opting for something that lives within NF Core, because NF Core is a community-driven effort, right? There's a higher chance of this pipeline that we want to use to be continuously developed, or at least monitored in the next few years. And then we also didn't want to reinvent the wheel. Variant calling is something that has been around for quite some time now. So we decided to utilize the existing variant calling pipeline, which is the NF Core sarek pipeline, where you can see the nice logo here. From the surveys that we conducted from people working within the PM4 Onco project, we know that all of these people have different backgrounds and preferences. And thus we decided to not only offer a reference pipeline written in Nextflow, but also one written in SnakeMake. And what we want is that both pipelines offer at least one route through all of the steps of the pipeline that have then the same tools available. So we want to have comparability. They don't have to be completely similar, right? But they just need to offer one route, one way of analyzing the data that is similar. But that also meant that we had to think about ways to alter both of these pipelines. And I will obviously talk a bit more about the sarek route, because that was my part of the project. And working on an NF Core pipeline means getting involved with the NF Core community, because obviously you could just fork the pipeline and just do whatever changes you want locally, but that's not what we wanted to do. We wanted to do something that also then came back to the community, because we didn't only want to take, we also wanted to give back, right? And for me, getting involved, my first step was working a lot on the modules repository. So, for example, porting modules to NF Test. And that was quite tedious, I must say, but it was a really good way of getting involved. So if you start working on the modules repository, you can do a lot of small PRs, you get a lot of engagement with different people across NF Core, you get used to the coding style, you understand the building blocks better. And what I did as someone who wanted to then at some point start working on the sarek pipeline, I picked modules that were used by sarek to work on them. I don't know, do some updates, do the porting to this new testing framework and so on. So I can really recommend trying to start there, because it's really small. It's not long lines of code. You can really understand it quite fast, I think. And then obviously the next step was to get involved with sarek, so joining the dev channel and then also the developer meetings, which are taking place on Mondays. And this is also a time for a quick shout out to Rick and Maxime. It was really nice, or it is still really nice to work with them, and it wouldn't have been possible without them helping me a lot and discussing things with me. And what we did in the developer meetings, for me, that was the possibility to discuss also what kind of tools and features we wanted after discussions in the PM4 Onco project, right? And it also helped me to understand how to implement these new features while also catering to needs of other users of the sarek pipeline, and then also ultimately implement them in a way that didn't break the sarek pipeline. So I would always recommend talking to the main developers. It's really nice. They are most likely to be happy if somebody starts contributing more. And for me, that worked quite nicely. And now after explaining the project and also how we got involved, I will show you in a few slides what types of things we actually added to the pipeline and why we did that. So first step was a new variant caller. It's called Muse. It's a pipeline. It's a variant caller used for somatic variant calling. So where you have both a tumor and a normal sample. And it's a new caller. It hasn't been there before. It uses a Markov chain model for variant calling. And what is really nice is that it's specifically for somatic analysis. And if you look at this diagram, which shows all of the variant callers available in sarek, you can see that this is one of the only variant callers for small variants that is available only for the somatic case. And we just wanted to add it to have a bit more variety in the benchmarking study. It's not a variant caller that is really used a lot by other hospitals, but it's a nice variant caller, and yeah, we just wanted to see how it performs. The next tool that we added is Velociraptor. It's also a sort of variant caller, but on a technical level, it's used after another variant caller, and it re-evaluates sort of the called variants of the previous variant caller. A good combination, for example, is FreeBase. And it is able to identify a wide spectrum of genomic changes. It can identify small variants, but also structural variants. And behind its hood, it uses a statistical model, and it's really, really flexible, where you can give scenario files to this tool, which is also indicated in the documentation here in the second paragraph, where you can see that there's different flags that we added to the sarek pipeline for users to be able to add their own scenario files. And with these scenario files, you are able to work with different types of data. So, for example, formalin fixed samples that are used a lot in the clinical practice is something that Velociraptor can handle. And it also offers filtration methods and so on. And the motivation, besides it being a nice tool, is that we wanted to have feature comparability to the SnakeMake workflow, because the SnakeMake workflow that we decided to use also has this tool. And last but not least, we added GPU accelerated alignment, so Parabricks, FQ2BAM. This is a possibility to utilize the GPUs that are more and more popular, because we have a lot of machine learning things coming up, to utilize these GPUs when you run your pipeline. And I think there's been a talk at the last summit about NF Core and GPUs. So if you're interested more in that area, then I recommend you watch this one. But I just wanted to tell you that this is part of the sarek pipeline now. And this was done because it was sort of a pipeline user request, right? In the future, there will be whole genome sequencing coming a lot in Germany, and whole genomes are really large for humans. So pre-processing with GPU-based alignment just improves the pipeline speed, and that is really nice in the clinical practice. And with this, I just want to show you my acknowledgement slide a little. I'm really thankful to be working in Tübingen. It's a really nice town. I'm working here at the University Clinic and also with my colleagues at QWIC, which is really nice. I'm funded by the BMFTR via the PM4Onco project. And also a quick shout out to my collaborators from Essen, Johannes and Bianca, who I worked with a lot during the benchmarking, or I'm still working with them a lot during the benchmarking part of the project. And instead of a summary slide, I just want to show you the release notes of sarek 3.6.0, which we just released a couple of days ago. And there you can see the features that I just talked about, but also other features that other people have implemented. And I would just encourage you to give it a go, try it out, and let us know if there's anything else that you want, I guess, or get involved yourself. Thank you.